Hello everyone, Mr. Linder here. Let's talk about transport processes. So in complex organisms like humans, it's important to understand how molecules and ions move within the intracellular and extracellular compartments, as well as look at how molecules and ions move across plasma membranes. Now, when you talk about transport, you're really going to see two categories. You're going to see transport mechanisms that require ATP, and those are going to be active transport mechanisms. Examples of active transport are going to be primary active transport, secondary active transport, and then we'll get into concepts of endocytosis, exocytosis, and phagocytosis. Transport that does not require ATP is going to be things like osmosis and diffusion, facilitated diffusion. In previous videos, I showed you how to calculate concentrations, and that was important for our understanding of osmosis. If you know the concentration of solutions, you can determine the movement of water. Because really osmosis is just the diffusion of water, but it's dependent upon the concentrations of the solutions. And so it turns out that water moves from areas of low solute concentration to areas of high solute concentrations. And so if you can determine the concentrations of solutions, you can determine which direction osmosis will occur. As we look at membrane transport, it's important to understand that there can be movement within the compartment. So for example, if this is a cell, let's say we have some glucose, put some phosphate attached to it. So this is glucose phosphate. It's trapped within the cell. That glucose phosphate can move within the cell. So it can, for example, diffuse from areas of high concentration to areas of low concentration. And so that's movement within the intracellular compartment. But you can also have movement across the membranes. So when you have, let's say we have a blood vessel, and we're looking at glucose inside that blood vessel, that glucose needs to move through the extracellular space, and then it needs to move across the plasma membrane if it's going to enter into the cell. And so that type of transport is going to require some sort of specific process in order for glucose to move across the plasma membrane. There's another concept that we'll look at in later discussions, which is something called bulk flow. And with bulk flow, you're talking about movement through compartments using a pressure gradient. So the example would be moving blood through the cardiovascular system, moving respiratory gases in the respiratory system. So as blood flows through this blood vessel, you're using a pressure gradient. Blood is moving from high pressure to low pressure, and we would refer to that as bulk flow. But in this discussion, we want to focus on these transport mechanisms for molecules and ions that are more specific and really are mechanisms, for the most part, for moving across the plasma membrane. And the reason we want to understand that movement is because we use molecules and ions in so many different physiological mechanisms. Everything from action potentials to muscle contractions to cell-to-cell communication, we're going to see some form of transport process involved. And so we need to be able to understand how and why certain molecules or ions move. In order to understand transport more fully, it's important to understand the concept of the plasma membrane being selectively permeable. So when you look at, say, a lipid bilayer, so we're looking at, these are phospholipids here, and there's two of them here in a bilayer. This is the plasma membrane. When you look at the plasma membrane, the concept of the plasma membrane being selectively permeable is important to understand. Because what that really means is that the plasma membrane only lets some molecules or ions cross and others cannot. And so looking at the selective permeability is going to help us to understand why certain situations may have proteins involved in the transport process. Because it turns out that some things can't get across the plasma membrane unless they have some sort of protein embedded in the membrane to assist that transport. So oftentimes, when we look at the selective permeability of the plasma membrane, we focus on the idea of things being lipid soluble or not. If a molecule is lipid soluble, then it can readily pass through the plasma membrane. Like things dissolve in like things. So nonpolar molecules that are lipid soluble can pass directly through the plasma membrane. But there are situations where things are not lipid soluble. And so if you have a situation where you have a molecule or ion that is not lipid soluble, in order for it to get across the membrane, okay, it's going to have to move through some sort of protein channel or have assistance, okay, through a carrier protein to get across the plasma membrane. So permeable substances, if we use the term permeable, permeable simply means molecules that can pass through the plasma membrane because they are lipid soluble. And things that are impermeable are going to be things that can't readily pass through the plasma membrane, but they can become permeable. Okay? So they might be initially impermeable, but they can become permeable if you have a situation like this, where it can use some sort of protein carrier or protein channel to get through the membrane. So the complexities of movement is important to understand. So what things are freely movable across the plasma membrane? Well, usually gases. So when we look at things like oxygen, carbon dioxide, those are going to be small gases that are permeable to the plasma membrane. Anything that is lipid soluble, okay, so anything that is lipid based, okay, is going to fall into that obviously lipid soluble category. So your steroids, okay, steroids are lipids. So testosterone, estrogens. This is why you can, if you use a birth control patch, let's say you place a patch on your skin and there are estrogens and progesterone, these steroids that are embedded into that patch. If that's placed on your skin, then the hormones that are in that patch can cross through the membranes because they are lipid soluble. And so those estrogens, right, progesterone hormones can cross through the plasma membranes, and they can easily get into circulation, and then it can help regulate the reproductive cycle. Insulin, okay, different hormone that you're probably familiar with. Insulin is not lipid soluble, and so you can't just rub the insulin on your skin. Instead, insulin has to be injected in order to get that into circulation in the bloodstream. So understanding transport processes is going to help you oftentimes in administration of medications and drugs. Ions. Ions are things that have a charge associated with them, so sodium, potassium, okay, calcium. Ions are not going to be readily permeable to the plasma membrane. They're relatively impermeable. But I say it's relative because if you have a sodium channel, okay, so if this represents a sodium channel, then sodium can pass through that channel. Okay? So sodium can move through if that's a sodium channel. If this channel here is a potassium channel, then potassium can move through that channel. So ions are relatively impermeable, but if you have channels, then you can make them permeable. Polar molecules, okay, molecules that have polarity to them, large molecules, they're going to have a hard time, proteins are going to have a harder time getting across plasma membranes. So again, in order to make them permeable to the membrane, you're going to have to use some sort of mechanism to get them across. So the size of the molecule and the lipid solubility of the molecule is going to have a great influence on whether something is permeable to the plasma membrane. If you're small, okay, if you're lipophilic, that means lipid-loving, lipophilic, then you're probably going to have an easier time getting across the membrane. But if you're larger, if you're lipophobic, meaning you don't dissolve in the phospholipids, then you're going to have a harder time getting across the membrane, and you're going to need some sort of transport mechanism. So again, what kind of transport mechanisms are there? Well, there's the passive transport and the active transport. So it can be simply passive transport that doesn't require ATP, or active transport mechanisms that do require ATP. One of the most important mechanisms to take a look at is the mechanism of diffusion. Diffusion is a passive mechanism. This is where molecules or ions move from a high concentration to a low concentration by using kinetic energy. So in order for diffusion to occur, it's using the kinetic energy of molecules or ions. In this simple experiment, here we have a high concentration of potassium in one location of this agar plate. We have a high concentration of Congo red in another location of this agar plate. If you were allowing this to run for 90 minutes, okay, what you see is diffusion. Okay. What's happening is the potassium iodide is moving away from the high concentration to areas of lower concentration. The Congo red is moving from areas of high concentration to areas of low concentration. And so there's diffusion taking place for these two dyes through the agar plate. Why did one, though, dye move farther than the other? You can see the distance here. The potassium iodide has moved farther than the Congo red. Well, in this case, it has a lot to do with molecular size. The Congo red is larger, okay? So this is the larger dye, and so therefore it moves slower. Okay? So larger molecules or larger ions are going to move slower than smaller molecules, or if you're comparing it to ions, smaller ions. So the potassium here must have a smaller potassium iodide must have a smaller molecular weight than the larger Congo red, and so it's moving faster. And so the diffusion is faster. So the molecular size mattered in this particular experiment. So when you're looking at properties of diffusion, what are some things that you can consider? Number one, diffusion is passive. Okay? So there's no energy, no ATP, I shouldn't say no energy, but no ATP required for diffusion to take place. Instead, we're relying on kinetic energy of the molecules or ions. The second thing to consider is the chemical gradient. Diffusion happens from high concentration to low concentration. So we're moving down the concentration gradient. The third thing to consider, diffusion will take place until you reach equilibrium. Now, that's not to say that equilibrium is what we're shooting for in the body, but diffusion does occur until you reach equilibrium. So if you had a situation where you had a container and you had something dissolving in the liquid of that container, there's going to be diffusion throughout that liquid until you reach an equilibrium. Now, in the human body, though, we talk a lot about homeostasis. Homeostasis does not mean equilibrium. So in previous videos, I've talked about how you can have differences in concentration outside and inside. So for example, sodium's in high concentration outside of a cell and low concentration inside of a cell. The reason they're not diffusing, the reason sodium does not diffuse to equilibrium is because there's other transport mechanisms pumping sodium back out. It's an active transport mechanism. So we have homeostasis of sodium, but if we didn't spend ATP energy, then the sodium would diffuse to equilibrium, because that is a property of diffusion. But because we spend energy to maintain homeostasis, we don't see that equilibrium take place. So keep that in mind. Homeostasis is not equilibrium, but diffusion will run until it reaches equilibrium. Diffusion is rapid over short distances. It's going to be slow over long distances. So the shorter the distance, right? Again, if we have a cell and we're just looking at diffusion, let's say of sodium again across the membrane, if it's a short distance, then the diffusion is going to be fast. But if you were relying on diffusion to move from the digestive system to your foot, if you just strictly relied on diffusion, that's going to be a slow process. So diffusion is good over short distances, but it's going to take a lot longer over larger distances. So we rely on our circulatory system when we're trying to move things over longer distances. Diffusion relies on temperature. So temperature is a factor in diffusion. When you increase temperature, you get faster diffusion. Therefore, if you decrease temperature, then you're going to get slower diffusion. Okay? So keep that in mind when you're thinking about diffusion. Okay? Molecules or ions are going to have more kinetic energy when there's higher temperature, and they're going to have less kinetic energy when there's lower temperature. So think about that as a property of diffusion. Molecular weight and size, I showed you in that previous diagram that larger molecules are going to move slower than the smaller molecules. And so molecular size is a factor in diffusion. And then diffusion, I've talked about this before, diffusion can happen whether it's across a membrane or whether you're just in a compartment. So if you're just looking at intracellular movement, go back to that glucose with phosphate attached to it, you can have diffusion, okay, as long as you have a high concentration and a low concentration. So you can have diffusion within that compartment, and you can also have diffusion across the plasma membrane as well. Okay? So those are properties of diffusion. Now, when you get into the movement of ions, as we look at things in the nervous system in future videos, there's also something called electrical attraction as well. And so we get into this idea of electrochemical movement. Okay? So electrochemical is a new layer to consider. We talked about the chemical, high concentration to low concentration, but there's also electrical attraction and repulsion to consider when you're looking at ions. Since ions have a charge, opposites attract like things repel. And so electrical forces come into play that will affect the diffusion of an ion, not just the chemical concentration gradient. So keep that in mind for future videos. How do you measure then diffusion? To measure diffusion, we look at something called Fick's law. And so Fick's law is going to take into consideration the solubility of a particular molecule. It's going to look at the size of the molecule. It's going to look at concentration gradient. It's going to look at membrane surface area. So there's all these different factors that come into play when you're trying to take a look at how to sort of quantify diffusion. So it's known as Fick's law. To simplify Fick's law of diffusion, we could look at something like this. Okay, so this is an equation for Fick's law of diffusion, where it says the rate of diffusion, okay, so how quickly diffusion is going to take place, is proportional to surface area, concentration gradients, and membrane permeability. And so again, that membrane permeability takes into consideration, is this molecule lipid soluble? Okay. It also, though, is going to have to take into consideration at times, do we have protein channels or protein carriers that are making the molecular ion permeable as well. So there's these different things to consider. So as you take a look at Fick's law, think of it in terms of rate of diffusion depending upon the permeability. So are we looking at steroids? Are we looking at lipid-soluble molecules? Are we looking at gases? Are we looking at water? Oftentimes water is an exception and it has a high permeability to plasma membranes. And that's oftentimes because there are protein channels embedded in the membrane. So water is sort of this exception to the rule when you look at membrane permeability. It almost looks like it's following the same pattern of permeability that a steroid would, but it's because there's lots of these aquaporin channels in our plasma membranes. And then we also have to take into consideration the surface area. Larger surface areas are going to have more permeability. Okay, so the larger the surface area, okay, I should say that the higher the rate of diffusion, and again, oftentimes because it's related to overall permeability of the membrane. So the more square units you have of surface area, the faster the rate of diffusion is going to be. You see that in patients with emphysema. As you destroy and lose lung tissue and you lose overall surface area, you're going to see a decrease in the rate of diffusion of respiratory gases. So as you have a decrease in surface area, you have a decrease in rate of diffusion. So you want to try to apply a lot of these concepts to pathophysiology. Okay? So in terms of lung function, you want to have high surface area, right? Increased surface area, increased rates of diffusion. And when you're looking at oxygen and carbon dioxide transport, that's very important. So when you have patients that have decreased lung function, it could be surface area that's a factor in affecting the rate of diffusion. And that's where Fick's law comes into play. Membrane permeability, as I mentioned, when you look at membrane permeability, we are definitely looking at lipid solubility. The more lipid soluble, the better the permeability is. Okay? Molecular size, though, has an inverse proportionality. So bigger molecules are going to be less permeable. So the larger the molecule gets, the less permeable it becomes. It's harder for it to get across the membrane. So smaller molecules and having lipid solubility are going to improve your overall membrane permeability and therefore increase your rate of diffusion. They have a positive effect on Fick's law. So that's a way to quantify diffusion by looking at Fick's law. Another thing to consider with transport is this idea of using membrane proteins for transport. I said earlier that when you're looking at overall permeability, sometimes something is not permeable. Let's say an ion like sodium is not permeable to a cell unless you have a protein channel that's going to allow that sodium to pass through it. So sodium is relatively impermeable because it's an ion. Okay, and ions don't just cross the plasma membrane. But when you have protein channels, okay, then what you're doing is you're increasing the permeability. And so oftentimes it looks like something like sodium is just moving across the membrane easily because there's all these protein channels embedded within the plasma membrane that are allowing that sodium to move across rather freely. Okay? So something like a steroid that just passes through the cell membrane, let's say you're looking at estrogen and it's just passing through the plasma membrane, sodium starts to look a lot like that transport because of all the channels that you might have. Okay? Same thing for potassium. If you have a lot of potassium channels, then the potassium moves freely across the plasma membrane. So membrane proteins are important, right, in our understanding of transport processes. Now, proteins can be embedded. They can be integral proteins and go all the way across the plasma membrane, or they could just be peripheral proteins. And then you can look at the various functions of them. So we're focusing on membrane transport, but you have to remember that proteins in the plasma membrane are structural proteins. They're enzymes in the membrane. There are lots of receptors for various chemical signaling, or even involved in transport processes like receptor-mediated endocytosis. So membrane protein functionality is an important concept to study. So what do we mean by a structural protein? A structural protein in the membrane would basically be cytoskeletal kinds of things. A structural protein might be involved in cell junctions, tight junctions, gap junctions, those kinds of things. Membrane enzymes would be involved in metabolism. They might be catalyzing a reaction for chemical signaling. We'll look at something called adenylate cyclase later on in videos where that enzyme is embedded in the membrane and it's used to make something called cyclic AMP. Okay? We also have membrane receptors. Talked a little bit about that. So here's a receptor embedded in the plasma membrane. And that receptor is going to interact with, let's say, a neurotransmitter or a hormone, or it's going to bind to some ligand, okay? Some molecule or ion that you're trying to bring into the cell. And so sometimes we use receptor proteins strictly for communication. And then sometimes we use receptor proteins as a way to perform receptor-mediated endocytosis. But then we also use proteins for transport processes. So these are integral proteins embedded into the plasma membranes. This is the plasma membrane right here. And those proteins are being used for transport processes. So we talk a lot about channels and carriers when you talk about transport proteins. So what is a channel protein? Think of a channel protein really as a hole. Okay? So basically you have a hole through the plasma membrane. So here's the plasma membrane again. And we have a way to move something in or out of the cell through this particular tunnel, a pore, or this hole in the plasma membrane. So ions can move through, water can move through aquaporins. Those are channel proteins. There are hundreds of ion channels. Some ion channels are specific for just sodium or just potassium or just calcium. Some ion channels move multiple ions, so sodium and potassium can move through. So it just depends on the protein structure. There are ion channel, though, selectivity. It's based on the amino acid structure. And so based on the positive and negative charges of the amino acids, that will dictate whether a positive ion can move through or whether a negative ion can move through. So there is definitely ion channel selectivity. Some channel proteins are just open channels, like the one we were just looking at. So you just have this hole that's open to the inside and outside. Sometimes these are called leak channels. So you'll see that term used sometimes, leak channels. And then we have other channels that are gated. So the gated channels fall into different categories. Some gated channels are chemically gated, meaning they're going to open in response to a chemical signal. So maybe a neurotransmitter binds to that protein and causes that protein to open. Some channels are voltage gated, and so they're only going to open when you reach a certain voltage of the membrane. In the nervous system, we talk a lot about neurons being at negative 70 millivolts for their resting membrane potential. But if you change that, let's say you get to negative 55 millivolts, you have changed the voltage of the membrane, and that possibly can open a channel that was previously closed. So maybe it was closed, if we draw it like this, okay? It's closed at negative 70, but it's open at negative 55. Okay? And once you open it, then some ion can move through that particular channel. And then you have mechanically gated channels. In mechanically gated channels, some physical force is causing the channel to open or close. Excuse me. So that mechanical force maybe pops open a channel. Maybe it's temperature, thermal related, some sort of pressure, and that's going to open the channel and then cause ions or molecules to flow through it, and then you get the physiological process going. We'll pick up next time with carrier proteins and get into the concept of active transport. So I hope this helps. Take care.